In the first portion of this lecture, we talked a little bit about why it's difficult to study the genetics of human longevity, but that we are able to do it using a particular technique called genome-wide association studies, or GWAS. GWAS allows us to study many different complex traits, like aging and longevity. And the way that it does that is by comparing the genomes of a case group to the genomes of a control group, looking for differences between the two, and then identifying where those differences are on your chromosomes. And so when we are studying aging, we'll have a group of long-lived or older individuals like centenarians, and we'll compare them to the general population. We'll take genome sequences from all people who are long-lived and genome sequences from the general population and compare all of these sequences to each other to look for differences or variants. And these variants are what are known as SNPs, or single nucleotide polymorphisms. And we're basically just looking for things that are different between the longer-lived people and the general population. And once we find the differences or find these SNPs, we can make associations between these SNPs and the area in the chromosomes that might be involved in aging. And so what this is down here is called a Manhattan plot. It shows all of the chromosomes in humans, and then SNPs, or the variants, are actually shown as small dots here above, above this region, and any variation between the two will be indicated by kind of the distance away from the bottom of the axis. And so you can see a large amount of SNPs here that vary between older and younger people, or cases and controls, a large amount of SNPs here, as well as here. And once again, these SNPs can be associated with a region on a chromosome. And so, for example, all of these navy blue SNPs here are in a region on chromosome 19. But what's important to remember about GWAS is it doesn't help us identify the specific genes involved. It just gives us an idea of where to look. So we know there's a lot of differences between long-lived and general population. And there's a big difference between this area of chromosome 19 in these two groups. We don't know what the gene is, but we can start looking there and using the sequence of the human genome to figure that out. And in particular, this shows data from a GWAS study on the longevity of humans and looking for genes that actually contribute to that longevity using a technique just described. And what you can see is that there are SNPs on several regions of the chromosomes that have been implicated in extended longevity and that are different between long-lived people and the general population. And some of those SNPs I'm going to highlight here. Some of those regions I'm going to highlight here, and the genes within those regions can be seen written. And we're going to talk about these genes in the next several slides. So the first gene that has been implicated in many different GWAS studies of longevity and aging is a gene called ApoE, which encodes the apolipoprotein E protein, which is a protein that transports lipids and cholesterol within the blood. ApoE is located on chromosome 19, and it's a protein that basically binds up different types of cholesterol in the blood and transports it throughout the body. There are 3 different alleles, or different forms of ApoE gene. There's E2, E3, and E4. And there are kind of good and bad alleles, or forms of the ApoE gene. The E2 allele is what's known as kind of the good allele, or the good form. And E4 is what's known as the bad allele, or the bad form. And you can see the different structures of the resulting proteins of these different ApoE alleles. And so ApoE2 has a protein shaped like this, It has a large area that lipids can bind to, whereas ApoE4, or that bad allele, encodes a protein that actually ends up kind of tightly closed and doesn't allow a lot of lipid binding. What's interesting is that there's an association of the E4 allele of ApoE, or the bad allele, with a decrease in longevity, as well as an increased risk of Alzheimer's disease. And within the population, the most common allele is actually the E3 allele, which is the neutral one, right? And so not that many people have the good, or the E2 allele, and not that many people have the E4 as compared to the E3. And what you'll notice is that that's sort of the general frequency of these three alleles in a Caucasian or population. But the frequency of Alzheimer's disease is dramatically increased, especially compared to the portion of people who have the E4 allele. And so a lot of people who, especially are homozygous for the E4 allele, will ultimately develop Alzheimer's disease. And there are ongoing studies on the ApoE gene and how it actually increases the risk of Alzheimer's. In the same locus or location on chromosome 19, there is another gene called TOM40, which encodes a translocase of outer mitochondrial membrane 40 protein, so TOMM40. And this is just a membrane protein that acts as a channel and allows protein transport from the cytoplasm into the mitochondria. And so you can see that TOM complex here. And what's not clear is how TOM40 is implicated in aging and longevity. And some people have hypothesized that it's really only implicated because of its proximity to the ApoE locus. And so while there have been a lot of people who have looked into TOM40 after discovering this locus of possible age-related genes from the GWAS study, no one's really been able to find anything about TOM40. And so it's likely only becoming implicated because it's so close on chromosome 19 to ApoE. And ApoE, as we just spoke about, definitely does play a role in longevity, especially that E4 allele, which decreases longevity. Another gene that has been implicated in GWAS studies is a FOXO3A gene, which is on chromosome 6, and it encodes a FOXO transcription factor, which is something we've talked about a bit in the genetic model organisms. So DAF16 in C. elegans and D-FOXO in flies are homologous to this FOXO3A transcription factor in humans. And as I will remind you, FOXO3A, as well as DAF16 and D-FOXO, are activated in response to insulin signaling. And so insulin signaling can basically affect transcription via FOXO3A. And FOXO3A is also regulated by SIRT1, or SIRT1, which is the homolog of SIR2 in yeast and in worms. And so the involvement of FOXO3A in longevity in humans argues that there's this kind of conservation of the insulin signaling pathway and a decrease in insulin signaling from yeast all the way up through humans. And there are two alleles or forms of the FOXO3A gene. There's a T allele and a G allele. And what's interesting is that when they compare the genomes of older and younger people, we find a lot of the G alleles in older individuals. And people who are homozygous GG tend to live very long. And so there's currently some studies looking into why it is that the G allele contributes to aging or how it contributes. But once again, I just want to stress the importance of this conservation of insulin signaling and its relationship to longevity. Finally, one other gene that has been implicated in several GWAS studies is the ABO gene on chromosome 9. And what ABO gene does is it encodes a enzyme or glycosyltransferase that converts H antigen to A and B antigen on the outside of red blood cells. And more simply, it's sort of the enzyme that helps determine what your ABO blood type is. And so there's an enzyme that can cleave H antigen on the outside of red blood cells and convert it into either A or B. And then depending on which allele you have for the ABO gene, that will determine what antigens you express and ultimately what your blood type is. And so what you'll notice here is there are the four different blood groups listed: Group O, A, Group B, and Group AB. And this is just looking at the odds of having one of those particular blood groups or blood types. At a certain age. And what you'll notice is that it seems that the chances of having AB or B blood, particularly for females, decreases with age. And the same is true for males. So type B blood and the odds of having type B blood also decreases with age. And so it's possible that there's some relationship between blood type and how long you live. With people who have type B blood or at least one B allele for the ABO gene, living shorter or having a decreased longevity as compared to other blood types. But what's also important to note is that these three alleles, A, B, and O, for the ABO gene, have different frequencies in the population. And the B allele is the lowest in frequency. And so it's not totally clear that this decrease seen in older individuals has anything to do with longevity so much as it does with allele frequency of the B gene in humans. Finally, the last gene we're going to discuss is a CDK gene, or a cyclin-dependent kinase inhibitor, which is a protein that inhibits cyclin-dependent kinases, or CDKs, to help control the cell cycle and inhibit growth. This is a gene also on chromosome 9, and what CKIs do is they actually bind to and inhibit active CDK cyclin complexes to stop the cell cycle from continuing. And while we know that CDKN2B is implicated by GWAS studies in longevity, there's not really a good understanding of how it contributes and the mechanism behind that. And the same can be said of these two genes here, SH2B3 as well as ataxin-2, both of which are on chromosome 12. One encodes a protein that regulates cell signaling. The ataxin-2 gene encodes a protein that regulates mRNA translation. Both of these have been implicated by GWAS studies in longevity and aging, but there's not really any studies explaining how that might work. And what's important to note is that the genetics of longevity don't just have to do with protein coding genes. There's also microRNAs, as well as long-known non-coding RNAs, which can play a role in longevity as well. Both mRNAs of specific types and long-known coding RNAs have been shown to change with age. And so it's possible that the genetics of longevity are not just controlled by protein coding genes, but by these non-coding elements as well.